It is really a great pleasure to be here and see any old friends. Thanks for the kind invitation. My name is Tao Sun from Chinese Academy of Sciences. Today I'm going to present some of our efforts in whole body PET connectome analysis. First, please allow me to introduce the place that I work. I'm working in Shenzhen Institute of Advanced Technology, which is an institute under Chinese Academy of Sciences located in Shenzhen, the Great Bay Area in China. The center I'm working with is Pathological Research Center for Biomedical Imaging. As you can see from the name, it covers several major medical imaging systems. We have one PET-MR in store, but no total body PET. So the work I'm going to present below is mostly the results from a close collaboration with my clinical partners. Okay, so I assume everybody in this seminar know more or less the potential of the total body PET. It is driven by the need of the synthesis research. How to say that? Previously, we tend to break down the whole person or whole body into levels at multiple scale and perform analysis at each level. For imaging, we tend to break down further. For example, we tend to look at the single organ of interest or lesion of interest. But really, the health and the disease states of the human relies on the complex neuronal, vascular, and neural systems, such as during the aging process, the integration analysis is getting more and more important. Indeed, there is an urgent need for us to explore the fundamentals of the interconnected body. This will further allow us to investigate the potential intervention strategies. Unfortunately, conventional research are mostly performed in vitro for the animal studies. So the problem here is we lack proper tool to investigate the human in vivo. PET as an imaging tool has the great potential to do this. Conventional multiband PET scan with classic tracer FDG can already provide information of the whole body distribution of the glucose metabolism. The introduction of the total body scanner further provides us the opportunity to look at this in a dynamic way. Combining with non-FD tracer, it will not only provide us a different angle to see existing problems, but also open the door for many unimagined opportunities. In order to fully uncover the hidden patterns at the whole body level, besides hardware, we also need proper software to allow us to investigate the connections among organs, hence the cell, protein, and genes. This really motivated us to propose logical analysis for whole body PET imaging. So what is the current status in performing PET connectivity research? If you are familiar with MRI, you probably know what is connectivity in theory. Topology-based image analysis grounded in graph theory is a method already used to study brain. In this approach, each brain region is regarded as a node, and the physical or functional connections between brain regions are represented as edges. By analyzing these nodes and edges, it has become possible to identify important features such as crucial nodes, modules, or pathways. The identified local and global features then can be applied to the diagnosis and disease diagnosis and the development of the treatment strategies. Compared to the conventional analysis methods that rely on the spatial or temporal information, topology-based analysis offers a fresh new perspective. As I said, knowledge is the most common used imaging modality for constructing brain networks MRI. For example, by processing the diffusion tensor imaging data, it is possible to construct a structural connectivity network based on the fiber tracks. Similarly, by processing fMRI data, it is possible to construct a functional connectivity network based on the correlation of the both signals. Back to PET, pioneer works already did similar thing back to 1984, which attempt to assess the glucose metabolic network for selected brain regions in healthy adults. This is even far earlier than the invention of the MRI, so there is no reason that we should not reconsider to use it. But different from MRI, the limitation here is the topological analysis for PET only allow us to access the group level information. The example here would be to observe the connectivity difference between the healthy and the Alzheimer's disease subject groups. Here, a reference metabolic network was constructed using the scans from a control group by performing the partial Pearson's correlation analysis between the SUVs at each in the pair. The matrix nodes represent the brain region, while the edge represent the strength of the correlation between the nodes. But to analyze a single subject is difficult. This is because the path SUV measure is only an integration over a certain period of time, let's say 20 minutes, unlike fMRI, which measures a series of the 3D volumes over time, which allows to compute the correlation of the temporal signals. Here is a study based on the FDG path that reviews the group-level metabolic connectivity patterns. It shows a reduction in the patterns of the inter- and intra-hemispheric brain connections, which decrease as the severity of the disease increases. In another study, despite the methodology difference, the test-retest reproducibility of the FDG pattern is similar to that of the established MRI-based network. FDG even has a higher proportion of the significant connections than fMRI and T1 networks. These findings support FDG network as a measure of functional brain connectivity. Similar approaches can also be used to investigate topological features of the armory path and the top path imaging. Even for receptor binding, we could investigate the interregional relationships using PET and found their change after treatment in anxiety disorders. To summarize, conventional connectivity studies for PET are mostly for brain imaging, but we are interested in whole body research. Question would be how to obtain the functional abnormality from a system perspective. This brings the aim of this study is to propose a framework that can construct a network that reviews individual metabolic abnormality. We are starting from the FDG here. Note the framework should not be limited to the scanners with long axial field of view, such as U-Explorer, but also be applied on the conventional scan. In the following slides, I will show you this is feasible. In the specific implementation, a normalized modeling approach is used to incorporate the patients into the healthy reference group. This helps to construct individual abnormalities in response to the disease. Before we dive into details, I will first introduce the datasets in the original paper that we published in 2022. 24 healthy subjects were included. Now we have a total number of 90. Also, 12 additional scans for testing, including 10 with the lung cancer, 1 with the COVID-19, and 1 with the gastrointestinal bleeding. All scans were acquired on a U-Explorer scanner. Note only 50 to 60 minutes SUV images were used instead of the dynamic image. For segmentation, a total number of 18 ROIs were manually delineated in the original paper, including 11 key organs and 7 brain regions. Now, this segmentation process was implemented fully automatically. As I said, the conventional way to construct a connectivity network for PET is at a group level. This means one would need a group of the scans, but due to the high heterogeneity nature of the body metabolism, we are often interested in a condition of the single subject. So the question is, how can we model the connectivity for single subjects per scan image, just like for brain MRI scan? The way we did here is called normative modeling. In other words, the subjects was added into the control group, which forms a new group with n+1 subjects. Thus enable us to build up another group level connectivity matrix. This allows us to subject the two group level connectivity and obtain the difference matrix that represent the individual patient subject abnormality. Then we can compute and visualize the z-score for each matrix element. This map essentially describes the degree of the metabolic abnormality at the whole body level. Under normal conditions, the body network structure is stable and relatively linked. However, when the metabolism is altered, the network connection will be altered accordingly. The benefit to perform such a normative modeling approach is the capability to derive the individual level connectivity information. Before we apply this approach to patient datasets, we first need experiments to validate their reliability and their reproducibility. Three aspects to look at. First, we want to validate the homogeneity of the control group. Well, for this validation, we found out that there is this low intercept variabilities in controls with an average similarity coefficient of 0.921, and there also exists high reproducibility as revealed by our sampling process. And we also found that a number of control sets are not that demanding. In our case, we only need a total number of 24 control data sets. Second, we need to validate the heterogeneity of the patient group. In this case, is the lung cancer group. We found out that the strains of each of these cancer patients' network was significantly different from the healthy reference network. On the other hand, there exists high heterogeneity among these patient subjects network with a quite low similarity of 0.196. This can be visualized as these two patient networks are indeed different from the healthy control network, but they themselves are different too. Additionally, the connectivity strains that link to lung appears to be more capable of separating the control from the disease group as we measure. The third thing, we validate whether this method has a similar quantification effect as the conventional SUV. Here, the change of the SUV was quantified as the level of the deviation. We can see that on the left is subjects that discharged from the COVID-19, and then on the right is subjects with gastrointestinal bleeding with unknown reason. For these two individual subjects, the change of the SUV delta SUV were found to be significantly correlated with the network strains at all organs. This can be visualized in metabolic connectivity plots for these two subjects. In comparison to the control network, the networks on the left demonstrate denser connectivity and higher strains at relevant nodes, which corresponding to the abnormal uptake organs, and which was labeled with the red arrows in the SUV images in the middle. Conclusion so far, we propose a framework that was able to derive both system and organ level metabolic abnormality using whole bodies SUV image. Note that this is not a direct measure of the molecular connectivity like fMRI, but rather a network perturbation, a template perturbation approach. Although This current framework only requires static image, but it can be extended to parametric images should a dynamic scan was available. We consider it as a complementation to the conventional SUV measures from a network perspective, and potentially can be useful for assessing the systematic disease, which we will show later. On the other hand, there are still many questions remaining to be answered. We answer part of them, but there are others. For example, um, there is this other methods to derive the individual information. Here we adopt a network template perturbation approach, but if you have dynamic data available, you can, of course, compute the correlation between the dynamic signal directly. Well, also there are other options, mostly can be adopted from the brain imaging. Another issue would be computation of the similarity measure. Here we use Pearson's correlation, but it could be others. Also, how can we relate the PET network to other connecting network from fMRI? Another big issue is how can we integrate connectivity? Because we want to know what caused the disease in one region, but not just the correlation between the regions. And the last thing is, can we combine the deep learning with this topological representation? Here, I just want to demonstrate this is possible. Basically applied for brain study. We applied a convolutional network to build high dimensional network features from top hat scan data. With that, we will be able to achieve good staging and early detection results for Alzheimer's disease. In this study, we have a task of 4 subgroups division to separate seeing control, early onset, late onset, AD. We can have accuracy of 85.7%. We can also predict whether subject will convert from MCI to AD in 36 months with an accuracy of 91.7%. In theory, we could extend such analysis approach to whole body topological analysis should a particular disease diagnosis and prognosis was identified. After the methodology, we are questioning ourselves what are the suitable applications for this kind of analysis tool. Back to this slide. Remember that we are only at the first stage and to explore the fundamental science of the body system. So for that, we could use this tool to perform both the cross-sectional and the longitudinal study targeting on the systematic disease such as oncological staging and treatment, inflammatory and infection, metabolism, and or the immune disease. The ultra-low dose feature of the total body scanner also allows us to research in healthy subjects, for instance, investigating the aging process. I'm not covered too much, but rather these several represented ones in the following slides. For oncology, we know prior to initiate immune therapy, it is necessary to assess whether a patient is suitable for the immune therapy, such as PD-L1 or CD8 therapy. Whole body PET imaging is an ideal visualization tool for assessing the target therapies compared to the conventional blood markers. By evaluating the metabolic patterns of the entire immune system before treatment, we think it is possible to predict the progression of the immune therapy. Some work here, they already attempt to monitor the organ of interest such as spleen or bone marrow but it may not enough. Well, for healthy aging subjects, it would be very interesting to understand the mechanism of how the glucose, fatty acid, amino acids change along the aging process in tissues such as in your muscle, bone, and fat regions. In fact, a multiparametric pattern can be derived from whole body dynamic path imaging. This would bring an insight to potential targets for us to slow down the aging process. Of course, we don't have to look at the entire body connectivity, but we could only be interested in the subnetwork, such as the model that only contains 2 or 3 organs. For example, cardiovascular and neural system interact in complex ways in both directions. Therefore, it is reasonable to hypothesize the disturbance in the brain-heart axis will influence the disease progression, such as major arithmetic events. Researchers has already found out that decreased glucose metabolism in insular hippocampus and the amygdala was implemented were independent predictor of the uh, major arithmetic events. This also applies to metabolic network that constructed certain brain regions. However, this study acquired the brain and heart in separate imaging sessions, so we could imagine with total body pattern imaging them simultaneously. Another limitation is it is only performed group-wise. So the question is, can we further establish the brain-heart connection mapping for individual and use that for prediction of coronary or the acute heart disease? Okay, for brain and gut, we know that serotonin system both exists in gut and brain. In fact, important peripheral express targets such as dopamine receptors has been so far only studied with path in the brain or in focal peripheral regions only. Can we actually build up multiple neuronal receptor binding maps at the whole body level to investigate the bidirectional brain to gut mechanism? For example, pain is assumed to related to serotonin expression both in gut and brain, but lack of direct evidence in human. We can imagine to identify the target to regulate the specific targets at one end that could develop a new treatment strategy not only for the bowel disease but also for the neuronal disease such as depression, especially when we consider we have so many PET radioligands that expressed in the whole body levels. With that, I want to thank my team member and my colleagues, and also the clinical collaborations from the hospitals.